Impossible only means that you have not found the solution yet. Every revolution seems impossible at the beginning, and after it happens, it was inevitable. A pleasant afternoon to everyone present over here. Today's esteemed guest speaker is one of such well-known Indian scientists who has found solution to many unsolved impossible aspects of bioscience. He is motivation to millions of people around the world and whose entire life is an example of excellence, hard work, and dedication with high determination. With this note, I, Dr. Dwani Upadhyay, Assistant Professor, Department with a green smile, feeling honored to welcome our today's emeritus speaker, renowned Indian scientist, distinguished professor at Indian Institute of Science, Professor Dr. V. Nagaraja, sir, on behalf of Parul University. Sir, we are truly fortunate to have you with us today. I'm very pleased to welcome our most honorable Dr. Emin Patel, sir, Provost, Parul University. Sir, we are truly encouraged with your presence. I would like to also welcome our HOD, Dr. Girish Babuke, and all the faculties and my dear students to this distinct live webinar. I assure you all that experiencing this live webinar will enlighten your knowledge with experimental life and learning lessons. Now, I would like to humbly request our most honorable Dr. Emin Patel, Provost Parul University, to say a few words on this August occasion. Please, sir.
Ji, namaskar to one and all. It's really a pleasure for me to be the part of such inaugural function. Being a provost, I try to join almost all. So at least I can interact with the stalwarts like Nagarajan sir, as he is the one of the expert person today. And the topic on which he is going to speak is the TB tuberculosis. So really it is a matter of interest for me also. So sir, first I will welcome you on behalf of Parul University. Always I preferred that the senior person or stalwarts from any organization joining, I used to connect because in Parul University at a time so many activities are going on. Every day there are 10 webinars. So obviously my duty to connect wherever it is visible, I try to connect it. So welcome again you sir, and hope so that whatever your experience at IIS Bangalore and whatever you have learned that may be given to the student by means of slide show, I hope so that that will be much worth to that. My knowledge to TB is concerned, I can say that in old time when we were very young, we have seen that number of cases of TB within our village, and there it is said at that time it is not curable disease, and so many people have died due to tuberculosis. But when what I've heard, maybe true or wrong. So within the course of 9 months of its after detection of the TB, at what stage it is, if proper medicine is taken without making a gap of any day, I think it can be cured easily. That is what I learned, whatever it may be. But as you said that it is our old enemy, have since ancient. But nowadays a lot of rather so-called discovery and so-called medicines are available in the area of tuberculosis. So hope so that our all participants will learn whatever your experience or your case study, whatever you have done your experience. Especially Parul University is no doubt you may be aware of it, but we are coming up very fast, growing up very fast. And we are getting the admission of almost 15,000 in every year. So obviously we are a big university where there are 100 buildings are there, 31 colleges are there, 21 faculties are there, 12,000 students are staying in the campus, and people like me, which is no doubt having a long experience, because I used to be a vice chancellor from Gujarat University, one of the biggest state universities. The same time I was vice chancellor of Gujarat Technological University, and so many digitalization across the Gujarat in government of Gujarat. This was my initiative. After completing after superannuation from the government setup, I have joined this Parul University, and here also by this so many activity and people like you we connect through the online, it became very easy for me to learn something. So my best wishes to this seminar. Hope so that lot of deliberation will take place. Whatever input you will give, our student make it interactive or may ask some of the questions. I will not take more time because you are the expert. You are going to speak. So I again welcome you, sir, and hope that this webinar will have a great success. Over to you, sir. Thank you.
Thank you, sir. Thank you for your always boosting us with our enlightening words, sir. Our ambient speaker is one of such person who made many theories practically plausible. Although Honorable Professor Nagaraja sir does not need any introduction as he himself is an epitome of aspiration and proud of the country. I would like to take this opportunity to introduce our today's speaker. Professor Nagaraja is a professor, Department of Microbiology and Cell Biology, Indian Institute of Science, Bangalore. He had received his BSc and MSc from Bangalore University. He completed his PhD from Department of Microbiology and Cell Biology at IISc. He was a research associate at Biozentrum University of Basel, Switzerland, at, and at the Department of Biology University of Rochester, USA. He joined as an assistant professor in Center for Genetic Engineering IISc and was involved in setting up the department. He became an associate professor at Department of Microbiology and Cell Biology IISc and then served as professor and chairman of the department. Sir has been appointed president of Jawaharlal Nehru Center for Advanced Research Scientist by the Cabinet Committee of Appointments. He has been awarded with many prestigious awards and fellowships. He is a fellow of Indian National Science Academy, Indian Academy of Science, National Academy of Science, the World Academy of Science, and lifetime member of several science bodies as societies. Sir had made outstanding contributions in understanding the role of modulation of DNA structure in gene expression and unique properties of microbacteria. He worked on microbacterial topoisomerase, has laid the basis for new drug discovery, and for the same he has been awarded with Shanti Swarup Bhatnagar Prize, highest award in India in the field of science by Most Honorable Prime Minister of India. Sir had received Srinivasan Memorial Award, first Product Process and Technology Development Award of Department of Biotechnology, Government of India, J.C. Bose Fellowship, Honorary Professor Jawaharlal Nehru Center for Advanced Scientific Research, IISA Alumni Award for Excellence in Research, Renbexi Science Foundation Award, Dr. Nityanand Endowment Lecture Award of INSA, J.C. Bose Medal of Indian National Science Academy, the World Academy of Science Prize in Biology. He has been awarded with prestigious Sir Mr. Vishweshwaraya State Award by the Government of Karnataka, India. I feel privileged to invite Professor Dr. Nagarajan Sir to deliver his talk, enlightening us with his knowledge. Sir, we would be honored to know about the drug resistant TB and various ways to encounter it with your lifelong experiences. Please, sir.
Okay. Thank you. Thank you. Can you hear me?
Yes, sir. You are audible.
Yeah. Is it loud enough?
Yes, sir. Yes, sir.
And you can see my pointer?
Yes, sir.
Yes, sir.
Everything is fine, sir. You proceed.
So at the outset, let me thank the Provost Sir for his gracious presence and for his words of wisdom for the youngsters who are assembled here. And I must also thank Dhwani for arranging this meeting, uh, asking me to give a talk. But it looked like she was giving a talk by giving a long introduction. I don't know from where she got my credentials from. So, but I didn't just share it. But I think these days one does get the thing. I think a very flattering introduction. Thank you, Dhwani. But now I have a tough job of keeping you all awake at 1:30. Is after lunch a good time to take sleep, and unfortunately, no. In this kind of a communication, in this online, I won't be able to see who is sleeping. In a regular class of even 300, I have a reputation of catching people who are sleeping. But today, I hope I will try. I hope most of you will keep awake during this talk. And unlike what Provost said, TB is curable, but now it has come back with the. I mean, he's right that TB is curable. It is absolutely right, but now we have a much more serious problem with the TB, and I'm going to talk to you about those kind of things. We have drug-resistant TB, and also little bit about our efforts. I'm not going to overload with our research efforts, but being a researcher, I'm not a clinician. I have not done any such clinical study, but what we have done is some research. When I say we have done it, the research is done by my students. Most of them listed here are past students because I'm presenting the work done, finished, concluded. What's ongoing now? Maybe I will do it. I will deliver it if I get a chance to come to Pearl University and interact with the students there. And have, because I have done this all over India. I have been to Delhi University colleges, I've been to Hyderabad, I've been to other universities, not just online mode, even the regular mode earlier. And in Baroda, I have been to, in Gujarat, I have been to Nirma University and very frequent visitor to MS University, Baroda at one time. And also been to Sardar Patel University in Vallabh Vidyanagar. So Gujarat is not new to me, and I have enjoyed my visits. And I hope this online meeting would be a prelude, a starting point for our interactions. And I will be very happy to talk to students. And after all, as teachers, that's the only thing we can do. We can't be like industrialists setting up something, but talking to students is something which is very dear to me. Right. So I'm going to talk to you today about how to tackle resurgent drug-resistant tuberculosis. It's not that simple, though Government of India, after they won the election, the present government, 2019 declared they would like to eradicate TB by 2025, if not by 2030. That's a very wishful thinking, but having worked lifetime in this subject, I know it is not going to be easy, almost impossible. Why I say so? This lecture will tell you why I am saying so. But however, efforts are on, global efforts are on. Right now, we are all in the middle of a big pandemic, unprecedented pandemic. At the moment, it is killing more people than TB, but with the result of this pandemic, all the healthcare workers who paying attention, TB has become even worse at this moment. Every other disease situation is worst. I'm sure Provost sir knows this because all the healthcare workers are put their efforts are on treating COVID, dealing with the COVID. But millions die of tuberculosis even now every year. So you stay tuned to my rest of the presentation. And the students have done the work, and I have collaborators. I have funded by Department of Biotechnology and a European grant called More Medicines for TB. Right. So, as some of you like Provost sir and others would know, this is one of the ancient diseases. Though it was identified by Robert Koch in 1840s, in the early 19th century, it is a very ancient enemy of mankind. Egyptian mummies, 5,000-year-old mummies, had lesions of the TB. That means this is very, very old disease, not one of those new diseases. We end up seeing in newspapers new viruses all the time, not just the current pandemic, even other ones. But TB is perhaps one of the most ancient diseases evolved with the humans. I won't go into that more now, except to say more recently we have multiple drug-resistant TB, extensively drug-resistant TB, and more recently in Mumbai, about 2 years ago, total drug-resistant TB. What do you mean by that? What we mean by that is no drugs, multiple drug-resistant TB, is resistant for 2 to 3 frontline drugs. Extensive drug resistant TB is resistant to all the frontline drugs and few additional drugs. And total drug resistance, the TB means there is no drug which can be used to treat it. Okay, and stay tuned. I'm keep coming back to this. So now is that, see, we think tuberculosis is a lung disease. It is in lungs, and that's it. And people are coughing, and the sputum sample testing, etc. But TB bacteria, being one of the cleverest bacteria, infects every organ of the body. No organ is spared. So you have many brain TB. You have, of course, pulmonary TB is what is most well known, well studied. This is where the chest X-rays shows up the things. Then you have bone TB, skeletal TB. Then your stomach TB, stomach and intestinal TB. Then you have eye TB of the eye, TB in the lymph nodes, and this miliary TB can be, it can be in the skin, it can be anywhere. It's called extra pulmonary TBs grouped under miliary TB. Then you can have genitourinary TB, urinary tract, your kidneys. So you can see almost every organ is covered. There are other organs. I must tell you a short story right away. I know of cases where one of my colleagues in Indian Science had a ear infection and fever. No, nothing could be diagnosed. Finally, doctors gave rifampicin and another anti-TB drug. It got cured. So because doctors then suspected a unculturable TB in the ear, and he was cured. And similarly, a young doctor came to me at some time. They said, when we treat patients for bone marrow transplantation, why bone marrow transplantation is done, under what cases. The people who got the transplantation, they ended up getting high fever. Why? They didn't know. There was nothing. He wanted to work in my lab, wanted to show that there is TB. I said, yes, let us start thinking about it. But then, of course, he didn't come back because he realized experimental work in the lab is more tedious. But we discussed the possibility that actually it's a reactivation of a dormant silent TB. As I'm speaking, I want to tell all of you who assembled there, wake up time. TB bacteria is present in most of us. Almost all Indian population. We are exposed to TB, but we don't come down with the TB. We don't come down with the tuberculosis disease, but tuberculosis bacillus, microbacterium tuberculosis, we get infection because after all, we travel in public and somebody will be coughing, aerosols, it will be transmitted. But then our immune system, our defense system of the body is strong enough, and we get protection. Our immune system is good, and so bacteria goes into hiding. So most of us have a dormant bacteria. Otherwise, how do you explain a old lady in US who was, when she was young, she was traveling in India, not only US, in Europe, they ended up getting TB after they were 70, 75 because they were not exposed to anything. It was a reactivation of a silent TB. So please be not under the assumption that TB is not serious enough. So we have to have a healthy lifestyle, good quality life, so that our immune system is strong, so that such a TB activation doesn't take place. So here is a world map. More than 1/3 of the world population is infected with TB. Yearly new infections occur, about 1% of the population, about 20 million chronic cases, and 2 million associates, about 20 lakh deaths per year. And miliary tuberculosis, disseminated tuberculosis, makes up 10% of extrapulmonary cases. Is my arrow visible? Hello?
Yes, sir. Yes, sir. It's visible. Yes.
And am I audible enough?
Yes, sir. Yes, sir. Absolutely.
Right. So, TB is, what is the TB treatment? World Health Organization recommended the multi-drug therapy. Ladies and gentlemen, boys and girls, dear students, most of the time when you go to a doctor for pneumonia or diarrhea, you end up getting one drug, not two drugs. But in TB treatment, right from beginning, we have to give multi-drug therapy. I will show you what are the drugs right in next slide. But before that, this multi-drug therapy has to be done at least for 6 months. If you don't do it, and for other reasons, you end up getting MDR TB. Then it is even longer treatment. A drug-sensitive TB, drug-sensitive tuberculosis, you can treat it as the provost sir said in 6 months. But MDR TB, it is a different question. So what you do? Short course chemotherapy recommended by WHO, and it's in India. There are many places. There's many other states is very well implemented. There is something called DOTS, direct observed therapy. So initial 2 months, there are 4 drugs: rifampicin, INH, pyrazinamide, and ethambutol. Rifampicin, INH, isonucleic hydroside, pyrazinamide, and ethambutol. 4 drugs are given first 2 months. Then in the continuation phase, you have only rifampicin and INH, 4 months. This is called fully supervised dose. And you, as Professor said, there is a 95% cure rate for drug-sensitive tuberculosis at the end of 6 months, provided the patient takes the medicine continuously. And most of you sitting there, you're like me. When the doctor gives 7 days medicine, we end up stopping it after 3 days or 4 days because it doesn't taste good and you don't want it, you're recovered, whatnot. But how do you expect a poor patient? Most of these TB are poor people, laborers, underprivileged, malnutritioned. And how do you expect, and whatever their whatever their savings may not be in Gujarat, in the rest of the places, they go it for their liquid diet. You know what I mean by liquid diet. They have to have that to drink in the evening. So they don't have, of course, these days we give the medicine is given free in the DOTS. Drug treatment is free in most places. Yet, patients may just not use it. So the success rate of 95% cure rate, provided they follow this regimen. I hope I made this point clear. But if it is not followed, and for other reasons, which perhaps we will discuss later, then it becomes drug-resistant TB, DRTB, drug-resistant TB, multiple drug-resistant TB. 18 to 24 months minimum treatment, and you have to use different drugs. Second-line tuberculosis, these are called first-line tuberculosis drugs. Second-line tuberculosis drugs like aminoglycosides, fluoroquinolones, they become second-line drugs. But 18 to 24 months is called DOTS plus. The second line, but they are very badly tolerated, and cure rate is less than 50%, mind you. And this for MDR. XDR, cure rate 50%. So, now diagnosis. Again, chest X-ray. This is after the, you end up seeing the lesions in the X-ray when there's lung. But then, in our country, in many developing countries, there's a childhood TB, miliary TB, rampant. There are 5 to 10% of our population is infected with TB. You can't diagnose it chest X-ray. It is not in the lungs. Finally, it may go to lungs. And there are so many new diagnosis methods. They're good, but not 100% sensitivity. 60%, 70%. There are different ones. I'm not going to bore you with all those things. But there are so many newer ones, but still we can't diagnose every TB. Why? Because TB can infect different organs, and many times you can't comes to some other infections. Within a day or two, hospitals are able to identify which drug resistant Pseudomonas or Klebsiella or E. coli is causing the disease. But in TB, culturing takes time, but you will not see the culture in some of the extrapulmonary TBs, miliary TBs, right? One of the cleverest bacteria, in fact, is actually much more clever than any other bacteria I know of, and I know quite a few bacteria. I teach microbiology. Um, it's a tough bug, tough bacteria. It likes nothing better than to be ingested by the host defense cell macrophage. Its usual home. Macrophage, it makes home. Can you believe it? And inside the macrophages, there's a tug of war. There's a war between sometimes macrophages wins and that tuberculosis bacteria dies. But in some other case, tuberculosis bacteria wins. How it wins, we are going to see now. But because the bacteria is a tough organism, protected by a waxy lipid and complex array of polysaccharides, coat and an ability to counter and find a way of escape host immune and chemical attacks. It's amazing bacteria. So in the warfare against the host, it's been continuously going on. And infection of tuberculosis, establishment of balance, macrophage, T lymphocytes, B lymphocytes, they are all our defense cells, immune cells, as you know. What it happens is, when the TB bacteria is ingested, it comes to the lungs, it deposits there and forms something called granuloma. And what you see in blue inside are the multiplying bacteria. Why they are multiplying? They inside the macrophage, they manipulate the macrophage for their survival. I will have one more slide for that. Inside the macrophage, MTB senses different environmental cues and modulates its transcription, modulates its gene expression, and starts replicating. And as it subverts it. All these host cells support the formation of this kind of lesions called granuloma. So this, you don't see this like too long. This is the mouse granuloma, it's a human granuloma. The lesions in the lung, isolated lung tissues. This is very, very bad too. Right. So just quick look. So the alveolar macrophages, lung macrophages, they ingest the when some patient coughs, the bacteria comes in, and when the bacteria is in blue, so this is the infected macrophage. Then the surrounding macrophages become foamy, called foamy macrophages, because they are converted to help this. Then the lymphocytes, white blood cells, they are also brought in here. They all become a nice layer. They are all subverted from their function. And then this is the adult granuloma, where some of this tissue lyses. The bacteria is multiplying here, multiplies. Then there is so much bacteria, and when this bursts, you cough and free bacteria have gone out, and that's how you start disseminating TB, spreading the TB. And so what bacteria does inside the host? Inside the host, this is entire thing is a one macrophage cell. So bacteria is engulfed into a vacuole, but then there is inside the cell, inside the host macrophage, you have lysosomes. Lysosomes are organelles in the body. The students would know, in these lysosomes you have enzymes to destroy invading stuff. They will destroy. They have enzymes to degrade proteins, RNA, DNA, lipids. But usually when a virus infects or some other bacteria infects, this vacuole brings it to lysosome. These two fuse. When you fuse, all these enzymes target the bacteria or virus, chop it down, chop its proteins, stop its lipids, stop its nucleic acids, and these are completely destroyed. And then from there, the immune response starts. Because then you can see, immune response has the innate immunity, adaptive immunity, then the adaptive immunity starts. But then, in the case of mycobacteria, this fusion of the phagosome, bacteria and vacuole containing the bacteria is called phagosome. Phagosome lysosome fusion is prevented by MTB. I need 1 hour to explain what are the different mechanisms, how it prevents. But for this seminar, I'm just going to tell you that bacteria is so clever, it prevents this fusion, because if it fuses, it's the end of the bacteria. If it doesn't fuse, it is able to multiply, it come out of this phagosome, go to the neighboring cell, infect there, and create all kinds of problem to the host. I hope I made this point clear. Yeah. Do you need any more clarification, or can I proceed?
Yes, sir. Please proceed.
Okay. So what I was saying so far, 1/3 of the world population carries latent TB infection. We are all latent TB infected. No available drugs can kill latent TB, hiding TB. A major, okay, I should say, in this is an active TB, but in some cases, when the situation is not good, TB goes into hiding. That's called latent TB, as I said. And it is hiding in many places inside the body. Only few things we know at this point. No ever and no ever drugs can kill latent TB. A major impediment in curing and possibly eradicating tuberculosis. Mycobacterium tuberculosis becomes dormant and phenotypically drug resistant when it encounters multiple stresses within the host. When the host is having a good immune system, like you, like Dhwani, myself, and all of you assembled there, and our immune system is strong, and as a result, it doesn't multiply, it hides itself. But then if it hides, drugs can physiological state, and this latent TB dormancy models one can try to do in the lab, but they are not perfect. So that's why we don't know how to treat the latent TB. Drug, an in vitro model that is required for new model is required for developing it. So just to repeat what I said, it's extremely ancient bacteria. For those who are sleeping, this slide is good enough. If you have not listened to the earlier part, very ancient bacteria, one of the cleverest bacteria, has found so many ways to survive inside the host. So many ways, hundreds of ways. Can stay dormant inside a healthy individual. Very difficult to handle. Unlike E. coli, which you get a culture in few hours, you have to wait 10 days to get the culture. Its division time is once in 24 hours. The division, and you have to use biosafety level 3 P3 facility. My students work in P3 facility because it's very highly infectious. You can't work in the regular laboratory like other bacteria. And then there is emergence of multiple drug resistant, extensive drug resistant, and TDRs. One thing I need to say, which I didn't say, is there is a fantastic understanding between HIV, the AIDS virus, and TB bacteria. They have a good nexus, good understanding, good connection. HIV patients die of tuberculosis, not because of AIDS. AIDS makes their system weak, and then more than 50% of the AIDS patients die of tuberculosis. So now you can see how they like each other, two enemies of mankind. So I can, if anybody has questions, I can talk later about it. It's a global challenge now. Why global challenge? I hope you guys are listening. When I was young, when the provost sir was young, TB was a problem of India. TB was a problem of Brazil. TB was a problem in third world countries. It was not a problem in Sweden. It was not a problem in developed Western Europe. Not a problem in the US. But in the last 20-25 years, it has become a global problem. 15-20 years ago, in hospitals in New York, people died of TB. Healthcare workers, nurses died of TB. And a few years ago in Sweden, 8 people died of tuberculosis because of this emerging drug-resistant tuberculosis. And so now there is a global concern about TB. So the global grand challenge, Bill and Gates Foundation has given money, and the pharmaceutical industries were not interested in developing new drugs for TB simply because there was no money, because it's all in the poor countries. But then now situation has changed. Now the industries are interested because of this resurgence of TB. So my job as a researcher is to find strategies to counter resurgent TB, and that is a part of the talk. But I'm going to give very little of our own work, because it's important for you people to know about the TB first. TB, the gravity of TB, because most of you won't think TB is such an important organism. Everybody knows SARS virus is important. COVID disease is very dangerous, but TB is actually very, very dangerous. And in fact, because our attention is on SARS, which is correct, which is right, and this is totally ignored at this moment. And I know of real struggle of people who are suffering with TB. So, how do you deal with TB? This is my own slide. In a sense, I thought about it and prepared it for this talk. So, pursue high quality dots expansion and enhancement. Address TB, HIV, MDR-TB and the needs of poor and vulnerable populations. Contribute to health system strengthening based on primary health care, and engage all care providers, and empower people with the TB and communities through partnership. Each one of these programs is not that straightforward, but in India, TB centers before COVID, they did a terrific job, at least in many states. I don't know how it is in Gujarat, but I know it's very good in all the 4 South Indian states. So where do I come? Where do the researchers come? Enable and promote research. So in the next few slides, I'm going to talk about the research, but I will finish in time, hopefully. But before that, how do you treat TB? You treat TB either by vaccine treatment or drugs or both. All of us got BCG vaccination. What is BCG? BCG is nothing but tubercle bacillus, which has been attenuated, it has been inactivated. It is done by none other than Pasteur. Pasteur gave the idea, and Calmette and Guerin developed it. That's why it's called BCG vaccine, Bacillus Calmette Guerin. Okay, so BCG vaccine we all get, but the efficacy of BCG vaccine is not very good. Especially in Indian population, it is not as effective. I think it's more effective in the Caucasians, the white people, but still it's not it's not the best. So world over, there are so many efforts to develop new vaccines and different kinds of vaccine to prevent infection, conventional protective vaccine, or boosting vaccine, which you can give bigger, better, longer lasting memory, booster doses, and then, or different therapeutic vaccine, or both vaccine which facilitates chemotherapy, drug treatment, and vaccine to prevent reactivation. There are efforts in all these directions by world over, and in fact, I'm not going to go into the list. This is only a partial list of various vaccine development. There is a Indian microbacteria called Mycobacterium indicus pranae, developed by Professor Pran Talwar, previous director of Indian National Institute of Immunology, and that is being tried out. And there are many others, including recombinant BCG, and this is only a small list. There is much more of it, and many of them are likely to be successful. But that's not good enough. Why it is not good enough? Because TB vaccines are not going to be fully effective. Because TB vaccines are widely used in Europe and elsewhere, seem to offer some protection from the disease, but have proved a dismal failure in developing countries due to differences in MTB strains. There are so many different kinds of M tuberculosis. There is called Beijing strain. There is called some other strain. So many. and there's a big difference in them. So vaccine efficacy comes into picture. Other co-infections, like if you have infected with another disease, then it becomes ineffective. Then nutritional genetic things and previous exposure to environmental bacteria. So extensive work on new vaccine approaches supported by European Union and others, but it will take 2 generations to attain success, if indeed this can be achieved. Thus, a continuing need for new drugs to treat TB, and particularly ones that are effective against currently virulent and mutant strains, multiple drug resistant variants, is an urgent need of the hour. So, Dhwani, I can go. Is it okay? The speed I went.
Yes, sir.
Okay.
Yes, sir. Yes, sir.
Right. So, why do you need new TB drugs? To shorten the total duration treatment. TB treatment is minimum 6 months, as I said in the beginning. But drug resistant TB 18 to 24 months. So, you need fast acting drugs. Remember young children, young students there. If you study well, Parul University, I believe, gives good education. Try to do research. Try to study well. And once you study, take up a major problem. And if you solve this problem, I don't think I will be able to solve the problem. TB is a bigger challenge. If you are able to find a new drug in which a treatment can be reduced from 6 months to 2 months, not 1 week, you will get Nobel Prize. From 6 months to 2 months, but that's a very big challenge. I said it in one sentence, but that's a challenge for you. If I'm alive, I'll help you, whatever I can, but with my knowledge. But but when you go to do research, I'm not sure how long it will be. But I think this is a challenge to shorten the total duration of treatment. And emergence of drug-resistant tuberculosis, another big challenge. We already discussed this again and again. TE, MDR, XDR, TDR, and treatment against latent TB infection. It's impossible at this moment. There is no way the drugs can go, because we don't know where are the bacteria hiding in the body. And slow progress in vaccine treatment. Vaccine treatment, even if you get successful vaccine, it will take 20 years. So we need urgently TB drug now. Okay. So the TB, the BCG is 85-year-old, the others, new ones are coming, but by the time many of them will not come to the final stage, so you need the drug. So in the last 15-20 years, people have woken up. Worldwide, there is effort to develop new drugs. This top one is a effort from Johnson & Johnson, Janssen Pharmaceutica in Belgium. They found this drug. Now it is used. It is fast forwarded. It is called bedaquiline. Clinically, it is used as bedaquiline. It is used for drug-resistant TB, even in India. But in India, it's very little used. But in the United States, elsewhere, in Africa, this bedaquiline is used for as for a drug-resistant TB treatment to bring down the time. This drug was developed by a European Union consortium, benzothiazones, called New Medicines for TB. Look at the number, headed by Stuart Core. New Medicines for TB. And even this is getting into phase 2 or phase 3 more. And there is a huge list of other things. Some of them have come to the come out clinically now, but I don't have time to explain this. But suffice to say, now there is more and more interest to get into TB research. And this European consortium, this is called New Medicines for TB. But in the next consortium, they invited me. I'm the only one from in India for the more medicines for TB. 28 groups from the world. One from India, it's me. Several pharmaceutical companies, AstraZeneca, and AstraZeneca, then Glasgow, so many other companies, so many universities from some 15 countries. We were part of that effort. So we contributed and developed some new drug there. Right. So I will come to that point in a minute. So how do you target microbacteria? It's a very tough bacteria. So our idea is to target its essential pathway. Think of a fort. The people inside the fort would like to protect themselves, and that's what mycobacteria does. So you have to try and find its weak point. Weak points are its enzymes, which are essential for mycobacteria, which is essential for its multiplication, which is essential for its DNA replication, which is essential for its RNA synthesis. RNA, okay, so we target this essential function in my lab. So they not only target, but it has to function as essential processes. They can be main regulators of the essential processes, and specific for bacteria, and it should be TB specific, not found in other bacteria, or if it is found, there's a difference in the properties and unique to the pathogen, different characteristics from the host enzyme. So we think of all those things, and we target this kind of proteins. I will not elaborate because these are all hardcore molecular biology. If somebody has questions, I will answer. So, when Prime Minister Modi ji visited the institute, I was asked to give a slide. Here is a this is a slide I presented. Mycobacterium tuberculosis, an ancient enemy of mankind, global one killer, emergence of multiple extensive diagnosis of TB, our strategy to combat the global health menace, targeting. So we developed the inhibitors against enzyme DNA gyrase, which I patented, and more work is going on. Development of first set of inhibitors for Topoisomer 1, another enzyme. I'll discuss a little bit about it. Then participation in European consortium to develop new drugs. I'll also discuss some of it. Right. So in my lab, we work on many enzymes. We do biochemistry, we do microbiology, we do molecular microbiology, we do cell biology, we do structural biology, we do a little bit of immunology. With the result, we can my PhD students, I only do the talking. They are very versatile. Very versatile. They can do many experiments, work day and night to produce results. So we work on all these enzymes. We work on enzymes called DNA topoisomerases. They are DNA gyrase and topoisomerase 1. I will explain them what they are. And we work on genetic switch, genes turning on and off. The transcription, gene synthesis is done by enzyme. DNA to RNA in the central dogma is done by enzyme RNA polymerase. And transcription is terminated by factor Rho. Then we work on the other transcription factors. So basically work on all the essential enzymes, essential processes in the pathogen. Because if we inhibit these essential processes, these processes. So I'm not going to, in the next 10 minutes, I'm going to stop this so that I'm well within the time, not more than 10 minutes. So the DNA, I'm going to explain only 2 or 3 cases, nothing more, because you don't need more. DNA inside all organisms is highly coiled. It gets highly condensed. But you can, in the electron microscope, you can see the open DNA of a small plasmid DNA. Otherwise, it is highly super coiled. It gets more coiled. It gets packaged into cell. You know, right? And this job is done by only one group of enzymes called topoisomerases. It's a completely tangled. Amazingly, cells do the equivalent of this by supercoiling of the DNA. Supercoiling is a smart form of compact storage of allowing easy manipulation. Otherwise, the DNA length is much longer than the cell. You read about it. So what do we do in bacteria? Bacteria chromosome is called as nucleoid. This is a nucleoid. And it's a textbook picture of when the cell is opened, the DNA spread out like a nice, beautiful flower. There's a DNA threads spread out. Otherwise, it is much more compact. Otherwise, which is actually more compact, as shown here. So, this nucleoid bacterial chromosome is organized by catalytic action of enzymes called topoisomerases and by decoratory bindings, which are not catalytic. They just bind to the DNA. They are originally called histone-like proteins. You would know about chromosomal higher organisms have histone proteins to bind to the chromosome. In bacteria, you don't have histones. You call them histone-like proteins or nucleoid-associated proteins. I hope I make it clear. So you have catalytic action of this class of enzymes, and these proteins which bind DNA, bend DNA, twist DNA, write the DNA, and together these two class of proteins package the DNA. Right. The topoisomers come in two different kinds. They make either single strand cut or double strand break, and these are textbook information. But they are like really magicians. They cut the DNA, pass the intact DNA across, and reseal the DNA. Cut the double strand DNA, pass the intact one, and reseal the DNA. As a result, you bring in extra coil or reduce, or if you go reverse, you remove the coil. So that's how the supercoiling is taken care of by these enzymes. And we work on this both type 1 and type 2 enzymes. And in the European Union Consortium, we developed a small molecule inhibitors called imiperimine and its derivative. But guess what? In 1950s, before any one of you are born, in TB sanatoriums in Switzerland, this was used as a drug. Why? Clinically, it is used as an antidepressant, but it worked for TB. But afterwards, people have forgotten. But we show in this European consortium that in the enzyme, this molecule docks nicely on topoisomerase 1. So in the neck, and this is called as repurposing of the old drug. And nowadays we are talking about repurposing the old drug. All the companies try to do this, and this is example of repurposing various chemicals as drugs. They are known to do something, but we end up using it for something else. So antidepressant drug, whether it can be used for TB is the question. Now, in fact, what we show here is, I will go quickly the data slide because these are all published in very big important journals. What you show is if it is blue, deep blue, it is surviving. Is red means is not surviving. When you treat with the drug, you end up seeing more cells dying, and in fact, several log killing of the bacteria is seen, both Mycobacterium tuberculosis and another mycobacterium called Mycobacterium phagosome. Right, and now we are trying to combine this. This is a discovery in that consortium. Now we are trying to combine this with other well-known drugs like Repurposing, INH. One of my students is working on it in the lab, trying to bring the combination. I got a big grant from DBT, sanctioned now finally, and we will be able to, next few years, I hope we should be able to develop the much better treatment combination. Let us pray. You all pray for us for our success. Okay. Now, just last 4 or 5 minutes, the nucleoid, I said, is the DNA is condensed by the action of these topoisomers. I just gave one example. We have drugs for other enzyme also, but I will not talk about it. Then also these histone-like proteins, nucleoid proteins. These are not enzymes, so you shouldn't be finding inhibitors. But we did something to find inhibitors. But what are these proteins? Nucleoid proteins. NAPs means don't take nap now, afternoon nap, please. Not for here. NAPs means nucleoid proteins. These are small proteins. They bind and organize genomes. They are involved in many DNA processes like recombination, repair, DNA synthesis, transcription, different steps of transcription. So they are extremely important to all the bacteria. And in mycobacteria, we show that one of the main protein called HU, Mycobacterium tuberculosis HU, is essential. So that is shown by genetic trick. When you reduce the level of this protein this is called Western blot protein levels. When you by genetic manipulation, which I will not explain, when you reduce the level of the protein, the cells will not survive. And when there is enough protein, the DNA is highly condensed, as you can see the blue. And when the protein becomes less, the DNA gets highly diffused. The nucleoid, now the DNA is spread out in the back, as you can see in the lower. See, here there is less protein, less of HU, DNA spread out. Why? And this is not good for the bacteria. The DNA has to thought maybe we should be able to develop a drug. Yes. So what? Okay. So when I'll skip. Okay. Basically, when you have too much of the protein, you can see a hypercondensed nucleoid. See, this is a nucleoid, blue staining here, highly condensed. This is normally the nucleoid is a little bit more spread, not as spread as the what is shown. It's highly condensed DNA is shown because you have expressed more protein by recombinant DNA. So this Tuhin Bhaumik, he was a MS University student. I went to give lectures in his institute. Then he said he wanted to come and do PhD in institute. Then I said, okay, try your luck. He came to institute, but he didn't get selected in my department. He was in the wait list, but then he got admitted in physics department. Though he was a MS University biotechnology student, he got admitted to physics department. There, my colleague Ram Kumar, he worked with him. I suggested him, you work with him, and then he can work with me. So he solved the structure of the protein, and we found compounds which can sit where DNA sits. Normally, DNA sits in this saddle. This is the structure of the protein. So when the compound sits here, DNA cannot bind. That's what he's showing here. This is the DNA shifting the free DNA, the protein-bound DNA. But when you add this drug, the DNA is displaced and the drug sits there. And as a result, you see what's happening is, this is the condensed nucleoid. When you have this inhibitor, see, nucleoid DNA is well spread, and in electron microscope you see condensed versus drug treated one spread out, and with the result the bacteria is dying, deformed, and several are killing. And we can do then RNA expression studies. When you do RNA expression, see, most of the genes are inhibited because the drug treatment has reduced the HU concentration, protein concentration, and we can validate by other results. And basically, how the cell is killed is, when you treat with the drug, the HU is cannot bind to DNA to do the various DNA transaction processes. So as a result, several genes are affected and cell gets killed. Okay, and in fact, this is showing so many genes are affected by treating with the drug. A small protein, not an enzyme, we treated with a small compound and we could kill the TB. And this also we are exploring further to take it along with the drug. Don't forget, TB is not easily treatable. You need 4 drugs, and these are not better than the existing ones. I must tell you, don't. But these are also, we are trying to see. We need to use the combination. With this combination, whether we can speed up. So last 2 slides, not data slides. I'm done with my presentation. So what is the wish? What is the wish of medical doctors? What is the wish of the researcher? What is the wish of the people who want to eradicate TB? You need a total bacterial sterilization. Sterilization means even the dormant TB should be cleared. And preferably oral delivery, not injection, because we prefer that is easiest. And it should be able to work on persisters, the intracellular persisting bacteria, dormant bacteria. And it should have novel mechanism of action so that it is active against multi-drug resistant and extensive drug resistant TB. And it should not be, see, sometimes you'll have 2-3 drugs. If you take one drug, will counter the others, so you'll have side effects. You have an antagonism. You don't want the antagonism with the DOTS treatment which I mentioned. So it has to be compatible dosing, compatible with the antiretroviral therapy, AIDS therapy, and antiretroviral therapy. You don't want to conflict there also. Toxicologically acceptable. Therapy ideally results in cure within less than 6 months, 4 months, 2 months. If 2 months would be a biggest miracle. So it's a big challenge. It's not an easy challenge, folks, ladies and gentlemen. But why TB, the barriers? Because not every company is interested. Governments put very little money. India has put some money, but that's not enough. Scientists don't get much money. Research money and market far less lucrative compared to cardiovascular, diabetic, and other health style diseases. reduced investment for TB. So many bigger companies have withdrawn, limited financial support, and progress in preclinical drug candidates from public sector. Very little happened. And the famous company Lupin, which is it patented joint patent with us, and afterwards they didn't take our product further because they're not interested, because it's a lot of work to take it the drug to drug, another drug in my case. Anyway, I think that's the end of my presentation, and thank you for your attention. Basically, I try to give you an overview. I try to give you an overview, not too much of my lab work, but I do much more intense lab work. I wanted to keep it as simple as possible, but nevertheless, some technical aspect is unavoidable. But I must say, I am not. It looks like I am rowing, but I am not rowing. I'm only speaking to you and speaking to other students elsewhere. That's my job, and all this great team, they're rowing the ship. Thank you for your attention.
Thank you so much, sir. Uh, I kindly request you, sir, to stop the presentation so that we can see you.
I have stopped it. Can you see it?
Yes, sir. Yes, sir. Now you are visible.
I hope.
Thank you so much, sir.
Yeah.
Yes, sir. Thank you so much. We are really very fortunate to hear you in person. I'm sure that all viewers are motivated, encouraged, and gained a lot of from your descriptive and illustrative session.
I,
Sir, respected sir, I actually request you that there are three questions what I have received from the students to ask you.
yeah. First of all, I went a little bit fast, but I'm not sure how. And it's not like a regular interaction face to face. But I'm happy to answer. I can stay here for next few hours. I think provost will have other work, but I think I can stay with the students if they want me to answer any questions.
Definitely, sir. We are very fortunate that you came on a very short notice, sir.
Yeah, right. So any questions I'm willing to
Yes,
take.
sir.
Yeah.
Yes, sir. Sir, we would like to know about your research experience while working on mycobacterial topoisomerase before discovery on new drug. So that's one student had asked me.
Uh, Say it again.
Yes, sir. He wanted to know about your research experience while working on mycobacterial topoisomerase before a discovering of new drug.
Oh, that's a very interesting question. Actually, so if since I think I thank you, dear student, who has asked this question. But I have some distractions here. Okay. Um, yeah, I started working on mycobacteria as a PhD student in Indian Institute of Science in 1975. Because historically in microbiology there was a lot of research going on in microbiology, cell biology department in Indian Institute of Science. So I started working in 1975. I started working on TB, but then after finishing, I worked on a bacterial virus. I worked on a bacteriophage which infects Mycobacterium spermatis, and for my PhD. So I started working that time. But last chapter in that was on enzyme called DNA gyrase, which is a type 2 topoisomer. That was discovered in 1976 by two scientists called Gellert and Cosarelli. So I was aware of it. My supervisor had gone to America for 18 months. So I thought of doing this work myself. So I showed that for phage multiplication in mycobacteria, you need DNA gyrase. That was one chapter in my thesis. Then I went to Switzerland, went to US, came back. When I came back, I saw not much work was done on tuberculosis, on gyrase, on topoisomerase. A lot of work in humans, a lot of work in E. coli, a lot of work in other bacteria, but not in tuber. So we did. So from that time we are working on these enzymes, topoisomerases. First, as I said, the student should realize you should understand the enzyme properties, how mycobacterial enzymes different from other enzymes. So initially students did that job. That led to that led for us to think of inhibiting the enzyme by new molecules. I hope my answer is okay.
Yes,
Yeah,
yes, sir.
yeah,
There is one more question.
yeah.
How can someone who sees you as an ideal can develop drug for such diseases?
Pardon me.
Yes, sir. The student named as Aman is asking, how can someone who sees you as an ideal can develop drug for such disease?
For what? Sleeping.
So, for the development of the drug for the disease, for
Which?
such diseases,
Okay, okay.
what
Yeah,
are
yeah.
going to?
No, no. First of all, I think you you should not take my case as ideal. In science, here, Aman, science, what you need to do is to study. See, when we are students, when the provost sir and myself were students, we had less opportunity. We had less opportunities, but we were fortunate also. But these days, there are plenty of opportunities, more opportunities, but more competition now. Much more competition than when we were students. So, but also you guys are smarter, but smartness goes in elsewhere. See, I hardly use mobile phone, but you people, perhaps some of you have two mobiles, and I end up seeing right now it's essential to have it. I check the myself contents.
Hello.
See, I'm not in Facebook, Twitter, any of those LinkedIn.
Hello.
I'm
Hello.
I'm not in any one of those ones. So I'm still passionate about my research. So if you want to become a researcher first, not a developing drug, you have to learn to climb the limitless ladder. What is limitless ladder? Learning science. Learning,
No. आता वेब पाहिजे नी. हा?
learn,
ते नोट कवर जाईल.
learning science. Learning science. There is no end. Okay. The
Yes,
more you learn, you climb. Okay. So you have to learn to understand that you have to do well in your subject. Then you can do PhD. Then you can do PhD and then yeah.
okay.
Okay.
Right, sir. Yes, sir. Yes, sir. And there is one last question, and that is from my side, sir.
Yes.
Students were very, very, very excited that you are going to speak in front of us. So, which advice would you like to give to our young students who are research enthusiastic and pursuing their UG and PG programs in bioscience?
Hello. तू बोलू शकते आवाज.
I didn't, I didn't hear you.
Yes, sir.
Yeah. Please tell
Sir,
me.
which advice would you like to give our research enthusiastic students?
As I said, no compromise on hard work. You have to study well. You have to read well. You have to interact with the teachers, and that is the way forward. And
Yes, sir.
and your basic strength, your basic ideas, fundamentals should be strong enough in the subjects. Then you can do well. Like I told you about your neighboring university, MS University student, biotechnology student, went to physics department to do PhD, and he did very well, published in a highly, very famous journal, and, with me also. So I think it's just a question of learning to do good. You have to be good at studies. As I said, there are more opportunities, but you have to grab the opportunities because there is more competition these days. You have to simply work hard. Your priorities, you have to set it right.
Yes, sir. Yes, sir. Yes, sir. Thank you so much, sir, for your for the sharing your deep knowledge with us. You are an epitome of inspiration to millions of citizens of India and abroad, and your entire life is a motivation to young science intellects. As despite so many odds and obstacles, you constantly work with high spirit and enthusiasm to achieve the goals and become high successful renowned scientist. Thank you so much, sir. I would like to request Provost, sir.
So unfortunately, my mic is not working. My mic is not working. There is a problem that mic is not working.
Okay, okay, sir. Okay. Yes, sir. So I would like to request you to kindly say few words to our amiable speakers, sir.
It's difficult for me to because my mic is not catching to my voice.
Sir, we are able to hear you. You are
Yes.
absolutely audible. Yes, sir.
Okay. I was under that impression that my mic is not working. That's why.
No,
Now
sir.
I
We
am.
can see you and we can hear you.
Thank you very much, sir. Nagarajan sir, as you have given a very good input, and I also learned little bit, but little bit the technical difficulty. What we can say that because we are operating on this digital phone, and sometimes there is a connection problem, sometimes there is a bandwidth problem, and so on. But also that this is a new era of learning which we have not seen in our time. But our now all students are very habituated to learn online in a better form than the offline. No doubt, offline cannot be substituted by online, but even the students are feeling very comfortable. And like that with the people like you, which normally we are inviting you, then due to the shortage of time or busy schedule, you may not come to our campus. But by this way, it became very easy for us to interact with people like you. So it's really a privilege for us, and also that in future also, whatever your knowledge you will share to our student will be benefit to us. I have visited twice IISc Bangalore when I was a principal LD College of Engineering for some of the TQ project. So I know that the campus and the working methodology of IISc Bangalore, that is a really beautiful academic institution where only thing is the learning. Even night time students are working in the laboratory. That's what I've seen. That is called the research. Otherwise, it is the stipulated time 9:30 to 4:30. Research is very difficult to be possible. But hope so that we are trying. We are also little restless campus. As I said, that 12,000 students are residing in the campus. So with that, I hope that with the guidance of people like you, we can do something in the research. Thank you very much, and with rather have a good day to you, and hope so that in future also we have some interaction. Thank you very much, sir.
Thank you.
Thank
Thank
you.
you.
Thank you so much, sir.
Bye.
Uh, yes,
Bye.
thank you.
Bye. Bye.
Thank you so much.
Bye.
Yes, sir.
I can leave now.
Yes.
Yes, sir. Yes, sir. Thank you. Thank you, Brijesh, sir. Although I have taken more time today.